Hello, and welcome to lecture number 6, unit 2. This is the first lecture in this unit, and the chapter is chapter 24 in Biology in Focus. This is for John Tyler Community College, Bio 102. My name is Mr. Sparks, and I will be guiding you through this lecture. Chapter 24, Early Life and the Diversification of Prokaryotes. Okay, the Earth formed about 4.6 billion years ago. It's believed that the solar system is older than that, and the universe itself is believed to be around 14 billion years old. So the formation of the Earth through gases and collecting debris occurred about 4.6 billion years ago. The oldest fossil organisms are the prokaryotes, organisms that lack a nuclear membrane or cellular organelles, dating back 3 to 3.5 billion years ago. So about a billion years passed when there was no life on Earth. Prokaryotes are single-celled organisms in the domains bacteria and archaea. Those are two of the three main domains. The three main domains are bacteria, archaea, and eukarya. Some of the earliest prokaryotic cells lived in dense mats that resembled stepping stones. These forms were called stromatolites, and they're seen here in this illustration of a artist's conception of what primitive Earth must have looked like. Here in the foreground here we see the stromatolites. These are bacterial mats that form in shallow, low oxygen environments. And it's likely that they were made up of cyanobacteria or photosynthetic bacteria that eventually liberated oxygen from the atmosphere and made conditions possible for life as we know it today. Prokaryotes are the most abundant organisms on Earth. There are more in a handful of fertile soil than number of people who have ever lived. So billions of prokaryotes. They are all over the place, and they have incredible roles to play in the ecosystem. Prokaryotes thrive almost everywhere, including places that are too acidic, salty, cold, or hot for most other organisms. So they have an incredible adaptability to extreme environments. Some prokaryotes colonize the bodies of other organisms. Okay, there's many bacteria. Staphylococcus is a common bacteria that occurs on the surface of the human body and other animals. It's normally not problematic. It's not a pathogen. In some cases, it does become mutated, and variant strains are detrimental to human health. Here's an example of bacteria on an agarose gel. You can see this is a handprint. Somebody just laid their hand on an agarose gel, and these are all of the prokaryotes, the bacteria principally, that grew from that handprint. So you can imagine bacteria are everywhere. Prokaryotes are all over the place. Conditions on early Earth made the origin of life possible. Chemical and physical processes on early Earth may have produced very simple cells through a sequence of stages. Abiotic synthesis of small organic molecules. Joining of these small molecules into macromolecules, and packaging of molecules into protocells or membrane-bound droplets that maintain a consistent internal chemistry. The origin of self-replicating molecules. So through these various stages, using theoretical biology and chemistry and physics, we're able to use the scientific method to deduce that this is the most likely path for life to have originated on Earth. Earth's early atmosphere likely contained water vapor and chemicals released by volcanic eruptions. This included nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, and hydrogen. As Earth cooled, water vapor condensed into oceans, and most of the hydrogen escaped into space. In the 1920s, Operin and Haldane hypothesized that early atmosphere was a reducing environment. This is an environment which would be more conducive to the production of certain chemistry than it exists in the atmosphere today. So today's atmospheric chemistry is not conducive to the occurrence of early biological related chemicals. In 1953, Stanley Miller and Harold Urey, this is one of my favorite experiments, conducted lab experiments that showed that abiotic synthesis of organic molecules in a reducing atmosphere was possible. What they did is they had a beaker with some piping, some glass, and they boiled the water in the beaker with the Bunsen burner. The water went up through the glass into a condensing unit, and beyond that condensing unit there was a cathode and an anode that created electric charges, and they zapped that primitive atmosphere with electric charges. And then in the collecting vase, they found primitive amino acids that suggested that these could be the basis of life. However, the evidence was not yet convincing, and the early atmosphere was in fact reducing. Instead of forming in the atmosphere, the first organic compounds may have been synthesized near volcanoes or deep-sea vents. The Miller-Urey type experiments demonstrate that organic molecules could have formed with various possible atmospheres. So they tried different, they used the Miller and Urey setup with different hypothetical atmospheres that may have existed in the past, and they got similar results for all of them. Organic molecules have also been found in meteorites, and this is a whole nother line of thinking. And so it's the possibility that organic molecules came to Earth on meteorites, and they were seeded on Earth as meteorites. Okay, here's in Miller and Urey's experiment. They collected these samples, and in these samples they measured them for various number of amino acids. And then later on in 2008, with improved chemistry, they went and they counted even more amino acids in the same samples. Here's an example of a volcanic eruption, and it causes lots of electrical activity. And this kind of high energy might have been necessary to create the amino acids in the Miller and Urey experiment and in the primitive Earth's atmosphere. RNA monomers, abiotic synthesis of macromolecules. So this is the synthesis of macromolecules through abiotic means. So there's without any life being involved, you can still synthesize organic macromolecules. RNA monomers have been produced spontaneously from simple molecules. Small organic molecules polymerize when they are concentrated on hot sand, clay, or rock. So it's possible that these RNA monomers were produced in the ancient atmosphere, and then they landed on clay or rock, and they produced even more complex organic molecules. Replication and metabolism are key properties of life and may have appeared together. Protocells, which are fluid-filled vesicles with a membrane-like structure, can also occur spontaneously. In water, lipids and other organic molecules can spontaneously form vesicles with a lipid bilayer. You can kind of think about if you put olive oil and vinegar together, that lipid layer creates these little bubbles. Well, that's similar to the lipids and water in this concept. Adding clay can increase the rate of vesicle formation. Vesicles, right, these little bubbles, exhibit simple reproduction and metabolism and maintain an internal chemical environment. So they maintain a type of homeostasis. Okay, when we look at, here are precursor molecules only. You take some of these precursor molecules, amino acids, RNA, and lipids, and you just don't treat them at all, then they do increase over time, but very slowly. But when you expose them to clays, a certain type of clay known as Montmorillonite clay is associated with volcanic explosions, and the primitive Earth conditions, there would have been a greater number of volcanic explosions. So this Montmorillonite clay would have been highly abundant. Okay, in those conditions, then these vesicles are more likely to occur. They increase in number. So, okay, here's a protocell vesicle right here. The vesicles can divide on their own, as in this vesicle, which is giving birth to smaller vesicles. And this vesicle has incorporated Montmorillonite clay particles, which are coated with RNA. So here's conceptually a primitive protocell. Okay, here shows the vesicles. The presence of Montmorillonite clay greatly increases the rate of vesicle self-assembly. The first genetic material was probably RNA, not DNA. RNA molecules are simpler, and they are called ribozymes and have been found to catalyze many different reactions. For example, ribozymes can make complementary copies of short stretches of RNA. Natural selection has produced self-replicating RNA molecules. RNA molecules that were more stable or replicated more quickly would have left the most descendant RNA molecules. The early genetic material might have formed an RNA world. So they were not really living organisms, but they had some of the traits. The protocells could duplicate, and within them they might have RNA, and that RNA required a homeostatic environment. And those conditions would have been favored by the ancient conditions. Vesicles with RNA capable of replication would have been protocells. RNA could have provided a template for DNA, a more stable genetic material. So RNA is basically the root chemical. And that would have given rise to DNA. And then, of course, we know through our studies in Bio 101 that DNA begets RNA begets protein, the central theory of genetics. Fossil evidence of early life. Many of the oldest fossils are stromatolites, layered rocks that form from the activities of prokaryotes up to 3.5 billion years ago. Remember, those are those stepping stone-like organisms that we looked at in the first artist's rendition of the primitive Earth's conditions. Atmosphere, fossils, and individual prokaryotic cells have also been discovered. Ancient fossils of individual prokaryotic cells have also been discovered. For example, fossilized prokaryotic cells have been found in 3.4 billion year old rocks from Australia. So those are ancient fossils. Okay, here are some of the oldest stromatolites dating back to 3.5 billion years old. Here are some non-photosynthetic bacteria. And here are other forms of free-living cyanobacteria. They all go back about 3.5 billion years old, with more of the fossils occurring in more recent times. And here's the stromatolites. You can see they form layer upon layer. Calcium carbonate gets deposited down upon one layer of the stromatolite, and they go on throughout time, throughout differing environmental conditions. They make layers upon layers. And those are fossilized, and then we can radiometrically date those fossils. Okay, stromatolites, non-photosynthetic bacteria, earliest possible earliest appearance in the fossil record. Okay, 3.5 billion year old cluster of non-photosynthetic bacteria. 1 billion year old fossilized stromatolite, and 1.5 billion year old fossilized cyanobacteria. The cyanobacteria that formed stromatolites were the main photosynthetic organisms for over 1 billion years. Early cyanobacteria began to release the oxygen into the Earth's atmosphere. Surviving prokaryotic lineages either avoided or adapted to the newly aerobic environment. Diverse structural and metabolic adaptations have evolved in prokaryotes. Most prokaryotes are unicellular, although some species form colonies. Most prokaryotic cells have diameters of 0.5 to 5 nanometers, which is much smaller than the 10 to 100 nanometers of many eukaryotic cells. So prokaryotes are much smaller. Prokaryotic cells have a variety of shapes. The three most common shapes are spheres or cossi, rods, bacilli, and spirals or spirochetes. Okay, so here's the spherical bacteria, rod-shaped bacteria, and spiral bacteria. Cell surface structures. A key feature of nearly all prokaryotic cells is their cell wall, which maintains cell shape, helps protect the cell, and prevents it from bursting in a hypotonic environment. Eukaryotic cell walls are made of cellulose or chitin. Bacterial cell walls contain peptidoglycan, a network of modified sugars that's cross-linked by polypeptides. This layer can help us identify different prokaryotes and classify them according to their nature. Archaeal cell walls contain polysaccharides and proteins but lack peptidoglycans. Scientists use the Gram stain to classify bacteria by cell wall composition. Gram-positive bacteria have simpler cell walls with a large amount of peptidoglycan. Gram-negative bacteria have less peptidoglycan and an outer membrane that can be toxic. Okay, so here's a conceptual understanding of gram-positive bacteria and gram-negative bacteria. So if you do a Gram stain on a bunch of bacteria and then you look at it under a high-power microscope, you can see that some of these bacteria are gram-positive. They accept that Gram stain, and some of these bacteria are gram-negative. There's two different types of bacteria here, and depending upon the conditions that you're looking at, the nature of whether it's gram-positive or gram-negative is going to be able to help you discern what type of bacteria you're working with in a given situation. Could be a health situation. Okay, gram-positive, there's the heavy peptidoglycan layer. There's the lack of peptidoglycan layer. More complex lipopolysaccharide. Many antibiotics target peptidoglycan and damage bacterial cell walls. Gram-negative bacteria are more likely to be antibiotic resistant. A polysaccharide or protein layer is called a capsule, and covers many prokaryotes. Okay, here's the bacterial capsule surrounding a spherical bacteria. This could be a streptococcus on the tonsil wall, on the wall of the human throat here. Here's a cell. Some bacteria develop resistant cells called endospores when they lack essential nutrient. And it's interesting to note that the Bacillus anthraci, the anthrax bacteria, goes into a dormant phase where it collects itself into an endospore, and it can last for hundreds of years. In fact, when archaeologists do digs on certain types of remains, like if they're looking at goat or sheep remains, goat and sheep remains are often associated with anthrax spores. They have to take special precautions because these anthrax bacteria can collect themselves into these endospores when they basically eliminate all the water from their shells, and they just remain in a desiccated condition until the conditions are proper for them to reassert themselves. Other bacteria have fimbriae, which are little kind of like furry hairs, which allow them to stick to their substrate or other individuals in a colony. Pili or sex pili are longer than fimbriae and allow prokaryotes to exchange DNA. So it's actually possible for prokaryotes to, they don't really reproduce sexually, but they can exchange genetic information through the sex pili. Here's the fimbriae on a bacteria, and it's just used to help collect the bacteria along with its like kind. Many bacteria are mobile. In a heterogeneous environment, many bacteria exhibit taxis, the ability to move toward or away from a given stimulus. Chemotaxis is the movement to or away from a chemical stimulus. So this is beneficial if they're moving towards a nutrient source. The bacteria can move towards that nutrient source using flagella, which help them move, or they can move away from toxin. If there's a toxin in their environment, the bacteria are actually capable of fleeing that toxic environment. Most motile bacteria propel themselves by flagella, scattered about on the surface or concentrated at one or both ends. Flagella of bacteria, Archaea, and eukaryotes are composed of different proteins and likely evolved independently. So even these different, bacteria, Archaea, and eukaryotes, they have it's like convergent evolution, where they have adapted the same solutions, but they did so in distinct pathways, distinct evolutionary pathways. Okay, here's an example of a flagellum. It's a pretty interesting organism. Many biologists believe that the filament of the flagella and the flagella itself is descended from the spirochete bacteria. So these could be spirochete bacteria that join up with a rod-shaped bacteria, and then they found it both to their advantage. This would be like a form of endosymbiosis, where the flagella would be part of the evolutionary pathway of the bacteria. Okay, here's a close-up look at the flagellum motor. Evolutionary origins of bacterial flagella. Bacterial flagella are composed of a motor, a hook, and a filament. Many of the flagella's proteins are modified versions of proteins that perform other tasks in bacteria. Flagella likely evolved as existing proteins were added to the ancestral secretory system. This is an example of exaptation. where existing structures take on new functions through descent with modification. So they could be related to the pili or the fimbriae, and then they evolved over time with different adaptations through the process of descent with modification. Okay, that's one pathway. Prokaryotic cells lack complex compartmentalization. Some prokaryotes do have specialized membranes that perform metabolic functions. These are usually infoldings of the plasma membrane. So here we have an aerobic prokaryote, which requires certain respiratory functions, and it has a respiratory membrane within the bacteria. So it's almost like an organelle. It's part of the organ of the bacteria. In photosynthetic prokaryote, it has these layers of membranes that function as thylakoid membranes. It's almost identical to a chloroplast. So in this respiratory membrane here is similar to a mitochondria. And this thylakoid membrane is similar to a chloroplast. Prokaryotic genome has less DNA than the eukaryotic genome. Most of the genome consists of circular chromosome. The chromosome is not surrounded by a membrane. It is located in the nucleoid region. Some species of bacteria have smaller rings of DNA called plasmids, and these plasmids are used to convert small amounts of genetic information, like the fertility factor that we're going to discuss in the future, and they're also useful to genetic engineers who can use the small amounts of DNA contained in the circular plasmids to transfer genetic traits from one organism to another through the bacteria. Here's a bacteria with its DNA expressed. This is all one chromosome. It's just scattered throughout. It's one circular chromosome. It's just scattered throughout the image here. And then here are plasmids, circular plasmids, little pieces of DNA that form the circular DNA independently. So here's one circular DNA, the chromosome of the bacteria, and here are plasmids, these circular rings of independent DNA. There are some differences between prokaryotes and eukaryotes in DNA replication, transcription, and translation. These allow people to use some antibiotics that inhibit bacterial growth without harming themselves. Nutritional and metabolic adaptations. Prokaryotes can be categorized by how they obtain energy and carbon. Phototrophs obtain energy from light. Chemoautotrophs obtain energy from chemicals. Autotrophs require CO2 as a carbon source. And heterotrophs require an organic nutrient to make organic compounds. Energy and carbon sources are combined to give four major modes of nutrition: photoautotrophy, chemoautotrophy, photoheterotrophy, and chemoheterotrophy. Okay, this is a table on page 465 in your textbook. I recommend you take a closer look at it. It describes the different forms of nutritional modes in prokaryotes. Photoautotrophs receive energy from light. They receive carbon from carbon dioxide, and these are photosynthetic prokaryotes like cyanobacteria, plants, certain protists, for example, algae. Chemoheterotrophs, they are able to get inorganic chemicals from their environment. These include like lithophiles or rock-dwelling prokaryotes, rock-dwelling Archaea. They obtain carbon dioxide from they contain carbon from carbon dioxide and other sources. They're unique to certain prokaryotes. Heterotrophs are photoheterotrophs that they receive energy from light, carbon sources from various organic compounds. These are like decomposers. They're unique to certain aquatic and salt-living prokaryotes. And chemoheterotrophs are organic compound. They receive energy from organic compounds. They get their carbon from different organic compounds. These include many prokaryotes and also protists, fungi, animals, and some other types. So these are the major nutritional modes, and they include descriptions of most of the prokaryotes fall into all of these categories. There can be prokaryotes that fit into any one of these categories. Other categories are distinct to plants, for example, the photoautotrophs, or animals like chemoheterotrophs and fungi. Prokaryotic metabolism varies with respect to oxygen. Obligate aerobes require oxygen for cellular respiration. Obligate anaerobes are poisoned by oxygen and fermentation or anaerobic respiration, in which substances other than oxygen require as electron acceptors. So there are some environments where you have obligate anaerobes. There's the Villa de Luz caverns in Mexico, which has a very low oxygen environment, and it has some of these obligate anaerobes, and they're believed to be among the most primitive organisms. Facultative anaerobes can survive with or without oxygen, so it doesn't really have an impact on their survivability. Nitrogen is an essential for the production of amino acids and nucleic acids. Prokaryotes can metabolize nitrogen in a variety of ways. In nitrogen fixation, some prokaryotes convert atmospheric nitrogen to ammonia. Okay, this nitrogen fixation is useful to different plant species. It helps those plants, those bacteria help those plants fix nitrogen and help them grow healthily. Cooperation between prokaryotes allows them to use environmental resources they could not use as individual cells. In the cyanobacterium Anabaena, photosynthetic cells and nitrogen-fixing cells called heterocysts or heterocytes are exchange metabolic products. So here's a strain of Anabaena. It's a bacterial chain, and here we have the photosynthetic cells, and then there are related non-photosynthetic cells that fix nitrogen, and they together the cells that fix nitrogen supply that nutrient to the photosynthetic cells, and the photosynthetic cells help supply other nutrients to the heterocysts. In some prokaryotic cells, metabolic cooperation occurs in surface coating colonies called biofilms. Now, biofilms are interesting. In fact, most of you are very familiar with biofilms. If you rub your tongue along your teeth, that coating that you feel there, that slick coating, is a bacterial coating of biofilm. Biofilms are also used in biological remediation, where a process where sanitation is pollutants are run through highly coordinated. Excuse me, I need to take a drink of water. So in biological remediation, you take ceramic balls and you coat them with bacteria, and then you run sewage through these, and the sewage helps to digest the bacteria, and out the other end you get clean water. Prokaryotes reproduce quickly by binary fission and can divide every 1 to 3 hours. This is part of their evolutionary success. Key features of prokaryotic biology allow them to divide quickly. They are small, they reproduce by binary fission, and they have short generation times. The adaptations of prokaryotes. Okay, an ongoing success of prokaryotes is an extraordinary example of physiological and metabolic diversification. They were here at the beginning of life on Earth, they're still here now, and they will be here long after human beings are gone. Prokaryotic diversification can be viewed as a first great wave of adaptive radiation in the evolutionary history of life. Rapid reproduction, mutation, and genetic recombination promote genetic diversity in prokaryotes. Prokaryotes have considerable genetic variation. 3 factors contribute to this genetic diversity. Rapid reproduction, we're already familiar with that. Bacteria grows very rapidly. It's why you get sick overnight. Mutation, bacteria are susceptible to normal rates of mutation. They're not exceptional, but because they reproduce so rapidly, any mutation that does occur is immediately duplicated and thrown into the gene pool. And genetic recombination. Genetic recombination is the potential for bacteria to take up DNA from its surrounding environment or from even another bacteria and to incorporate that DNA into its genome. Prokaryotes reproduce by binary fission, and offspring cells are generally identical. Mutation rates during binary fission are low, but because of rapid reproduction, mutations can accumulate rapidly in a population. A high diversity from mutations allows for rapid evolution. Prokaryotes are not primitive, but are highly evolved. Okay, so that's that's interesting because it makes you ask the question of what is primitive. So prokaryotes, they are an ancient lineage, but they're highly adaptable, and therefore they're highly evolved to fit in today's environmental situations. So they're ancient, but they're also not primitive. Okay, here's an experiment where Cooper and Lenski tested the ability of E. coli populations to adapt to a new environment. They established 12 populations, each founded by a single cell from an E. coli strain, and followed these populations for 20,000 generations, 3,000 days. That's a couple of years. That's like 3 or 4 years. No, it's longer than that. To maintain a continual supply of resources each day, the researchers performed serial transfer. They transferred 0.1 milliliter of each population to a new tube containing 9.9 milliliters of fresh growth medium. The growth medium used throughout the experiment provided a challenging environment that contained only low levels of glucose and other resources needed for growth. Okay, the fitness of the experimental populations as measured by the growth weight of each population increased rapidly for the first 5,000 generations, the first 2 years, and more slowly for the next 15,000 generations. So it shows that the growth shows that the averages of 12 populations. So this suggests that the bacteria were able to adapt to their new environment up to a certain condition. Once they had reached those conditions, then they no longer increased in their exponential rate of growth. Genetic recombination, the combining of DNA from 2 sources, contributes to diversity. Prokaryotic DNA from different individuals can be brought together by transformation, transduction, and conjugation. Movement of genes among individuals from different species is called horizontal gene transfer. And horizontal gene transfer is a great part of our evolutionary past in prokaryotes. A prokaryotic cell can take up and incorporate foreign DNA from the surrounding environment in a process called transformation. So prokaryotic cells can actually pick up strands of foreign DNA in its environment and incorporate them into its genome. Transduction is the movement of genes between bacteria and bacteriophages. So viruses that infect bacteria can introduce novel genes into the bacteria. Okay. Here, a phage infects bacteria donor cell with one type of gene and with A and B alleles. The phage DNA is replicated and proteins are synthesized. The fragment of DNA in the A allele is packaged within the phage capsid. Then the phage moves on to a new bacterial cell, and it infects that bacterial cell with the new gene. The incorporation of the phage DNA creates recombinant cell with genotype A and B. And as long as that bacteriophage no longer, if that bacteriophage does not cause that cell to lyse, then that cell can go and take that recombinant DNA on to its next binary fission. Conjugation is the process where genetic material is transferred between prokaryotic cells. In bacteria, DNA transfer is one way. The E. coli, the donor cell, attaches to the recipient by a peleus. The peleus pulls it closer and transfers the DNA. Okay, so here's a sex peleus. It's this one. This bacteria with all the fimbriae on it is the bacteria that's generating the sex peleus. It senses the location of the other bacteria, and it extends its sex peleus, and then it transfers DNA through this sex peleus into the new bacteria, and then that bacteria will then incorporate those traits into its chromosome, into its genome. The F factor is a piece of DNA that is required for the production of the peleae. The F factor stands for the fertility factor. It's the factor that causes the DNA in able to be produce the sex peleae. When it transfers, when it connects the sex peleae onto the other bacteria, it generally transfers that F factor into that new bacteria. So then that bacteria and all of its offspring can disperse its genetic code in this same way. Cells containing the F plasmid function as DNA donors during conjugation. Cells with the F factor, with the F minus factor, function as DNA recipients during the conjugation. Okay, so there's donors and recipients, and the F plasmid, F positive function is the donor, and the F minus is the recipient. The F factor is transferable during conjugation. So one bacteria can cause another to be the donor. Okay, so here's the F plasmid. Remember, the plasmid is this little circular chromosome-like piece of DNA in the bacteria. The bacterial here are the bacterial chromosome. Here, the bacterial chromosome are somewhat distinct. Here's the mating bridge, or the sex peleus. It transfers that F plasmid into the new bacteria, and that new bacteria then takes on that trait, that F plasmid trait. And it can also transfer bits of other DNA into that during that transfer. Okay, so one strand of F positive cell plasmid breaks at the arrowhead. This the broken strand peels off and enters the F-negative cell, the donor cell. The donor and recipient cells synthesize complementary DNA strands, and recipient cell is now a recombinant donor cell. The F factor can also be integrated into your. The F factor can also be integrated into the chromosome. A cell with the F factor built into its chromosomes function as a donor during conjugation. The recipient becomes a recombinant bacteria with DNA from two different cells. If you want to get some more information on this, check out your textbook on page 469. R plasmids and antibiotic resistance. Genes for antibiotic resistance are carried in R plasmids, and antibiotics kill sensitive bacteria, but not bacteria with specific R plasmids. Through natural selection, the fraction of bacteria with genes for resistance increases in a population exposed to antibiotics. Antibiotic-resistant strains of bacteria are becoming more and more common. So these R plasmids they do have an effect through natural selection. Prokaryotes have radiated into a diverse set of lineages. Prokaryotes have radiated extensively due to diverse structural and metabolic adaptations. Prokaryotes inhabit every environment known to support life. Everything from the Antarctic to the deep sea vents and in the deepest parts of the ocean, a mile down under the sea by the volcanic vents. Applying molecular systematics to the investigation of prokaryotic phylogeny has produced dramatic results. Molecular systematics lend to the splitting of prokaryotes into bacteria and archaea. Molecular systematics continue to work on the phylogeny of prokaryotes. So the molecular systematics is a way of understanding taxonomy and systematics, and it's added to our understanding, and we keep changing this understanding through time, especially with modern events and technologies. Okay, this is a phylogenetic tree based on molecular data, and it shows one of several debated hypotheses of the relationships between major prokaryotic groups discussed in this chapter. Within Archaea, the placement of the Prokaryotes and the nanoarchaeocytes remains unclear. But this is the universal ancestor. This is the most primitive organism, the most primitive prokaryotes, and then it evolved into these different domains here. We can see these domains have subdomains within them, many different types in the domain Archaea, and the domain bacteria includes these different supergroups. And this is the Archaea. This is the eukaryotes. So you can see the eukaryotes includes all the everything, the protists, everything from the protists all the way up to human beings. So it's all these organisms that are have a nuclear membrane and organelles, and you can see that the diversity is less in the eukaryotes than it is in the Archaea or the bacteria. The use of polymerase chain reaction, or PCR, has allowed for more rapid sequencing of prokaryote genomes. A handful of soil may contain 10,000 prokaryotic species. Horizontal gene transfer between prokaryotes obscures the root of the tree of life. Bacteria include the vast majority of prokaryotes familiar to most people. Diverse nutritional types are scattered among the major groups of bacteria. Okay, Eukarya, Archaea, bacteria. Here's a very simplified. This only includes the three domains. Okay, within those three domains, we look at the proteobacteria, which includes 1, 2, 3, 4, 5 different subgroups. These subgroups are they're pretty. There's a lot of information here, and I want to guide you to page 472. I'm not going to give you this information in this presentation because it's too involved for our purposes for this lecture today. But go and check out these proteobacteria. Proteobacteria are gram-native bacteria. Proteobacteria, and this is on page 472. Proteobacteria are gram-negative bacteria, including photoautotrophs, chemoautotrophs, and heterotrophs. Some are anaerobic, and others are aerobic. Okay, proteobacteria, alpha, beta, gamma, delta, epsilon. They're quite a diverse group of bacteria, all within the domain bacteria. Members of the subgroup alpha proteobacteria are closely associated with eukaryotic hosts in many cases. Scientists hypothesize that mitochondria evolved from aerobic alpha proteobacteria through endosymbiosis. Example, rhizobium, which forms root nodules in legumes and fixes atmospheric nitrogen. This is a very important leguminous vegetables like soybeans grow. They grow these. The bacteria gets into their roots and it helps them to fix atmospheric nitrogen and helps those. It's part of the nutrients that the plant needs to grow. Agrobacterium, which produces tumors in plants, is used in genetic engineering. I don't have a good understanding of this. Okay, rhizobium, inside the root cell of a legume, the alpha subgroup of the proteobacteria. Members of the subgroup beta proteobacteria are nutritionally diverse. The example include the soil bacterium nitrosomonas, which converts ammonium to nitrous oxide. Nitrosomonas here, the beta subgroup. The subgroup gamma proteobacteria includes sulfur bacteria such as Thiomargarita namibiensis and pathogens such as Legionella is responsible for Legionnaire's disease. Salmonella, I don't know if anyone's familiar with salmonella, but I've had salmonella. It's quite an uncomfortable disease. And Vibrio cholerae, which is the organism responsible for cholera. Escherichia coli, or E. coli, resides in the intestines of mammals and is not normally pathogenic. This is also a gamma proteobacteria. Here's the gamma subgroup, Thiomargarita containing sulfur waste. The subgroup delta proteobacteria includes the slime-secreting myxobacteria and Delavibrios, a bacteria that attacks other bacteria. The fruiting bodies of Chondromyces, a myxobacterium, in the delta subgroup. The subgroup epsilon proteobacteria contains many pathogens, including Campylobacter, which causes blood poisoning, and Heliobacter pylori, which causes stomach ulcers. Here's Heliobacter pylori. Some of you may be familiar with it. It causes stress. It can be induced by stress-related conditions to cause stomach ulcers. Okay, here are the rest of the bacteria: chlamydias, spirochetes, cyanobacteria, streptomyces, mycoplasmas. Chlamydias are familiar with you all. These parasites can survive only within animal cells, depending on their hosts for resources such as ATP. The gram-negative walls of chlamydia are unusual in that they lacked peptidoglycan. One species, Chlamydia trachomatis, is the most common cause of blindness in the world and also causes non-gonococcal urethritis. the most commonly sexually translated disease in the United States. The spirochetes, these helical gram-native heterotrophs spiral through their environment by means of rotating internal flagellum-like filaments. Many spirochetes are free-living, but others are notorious pathogenic parasites. Treponema palladium causes syphilis, and Borrelia burgdorferi causes Lyme disease. Cyanobacteria, these are nicer bacteria. They gram-negative photoautotrophs, so they only prokaryotes with plant-like oxygen-generating photosynthesis. In fact, chloroplasts likely evolved from endosymbiotic cyanobacterium. Both solitary and filamentous cyanobacteria are abundant and components of freshwater and marine phytoplankton. The collection of photosynthetic organisms that drift near the water surface. Some filaments have cells specialized for nitrogen fixation, the process that incorporates atmospheric nitrogen into inorganic compounds that can be used in the synthesis of amino acids and other organic molecules. And then there are the gram-negative bacteria, including the gram-positive bacteria. They rival the proteobacteria in diversity, and there's just lots of different types of them, including streptomyces, which is known to cause strep throat, and mycoplasms, which some of them are pathogens and others are not. Mycoplasms are the only bacteria known to lack cell walls. They're also the tiniest known cells, with diameters as small as 0.1 nanometers, about 5 times as large as a ribosome. Chlamydias are parasites that live within animal cells. Chlamydia tracheomitis causes blindness and non-gonococcal urethritis by sexual transmission. Chlamydia inside an animal cell. That's where the arrows indicate. Here are the chlamydias. Spirochetes are helical heterotrophs. Some are parasites, including Treponema palladium, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease. Spirochetes, these are the spirochetes, the Lyme disease spirochetes. Cyanobacteria are photoautotrophs that generate oxygen. Plant chloroplasts likely evolved from cyanobacteria by the process of endosymbiosis. Here are the cyanobacteria. You can see in their green color that indicates they're chloroplasts. Gram-positive bacteria includes the actinomycetes, which decompose soil, streptomyces, which are a source of antibiotics, Bacillus anthracis, the cause of anthrax. Remember, the anthrax forms the endospore, which can exist for hundreds of years in the soil. Clostridium botulinum, which causes botulism. Botulism is one of the most deadliest poisons in the world. It's also the compound used in Botox injections. So when people get plastic surgery, they use one of the most deadliest toxins in the world, botulinum toxin. Some Staphylococcus and Streptococcus, which can be pathogenic, and mycoplasms, which are the smallest known cells. Here's the streptomyces, and here's mycoplasms. They're covering a fibroblast, human fibroblast cell. There's tiny little cells scattered all over here. These are all the mycoplasms. Okay, now we're talking about Archaea, which are an interesting group of prokaryotes. Archaea are in their own domain, and they share certain traits with bacteria and other traits with eukaryotes. So bacteria are off on one branch here, and Archaea and eukarya are off. They have a common ancestor back here. So we can tell through certain characteristics of the ribosomal production in Archaea, that they are more closely related to eukarya than they are to bacteria. But they're prokaryotes, so they share that characteristic with bacteria. Okay, here's a comparison table of the 3 domains of life. That is on, let's see. Let me try to find that for you. Okay, on page 471, you'll find this table, the comparison of the 3 domains of life. I'm not going to go through this. I'm going to go through this very quickly. Nuclear envelope, absent in Archaea, absent in bacteria, present in eukarya. And some species of eukarya are able to grow at temperatures above 100 degrees Celsius. So there's just an incredible range throughout here. And I urge you to go and take a look at this table on page 471. So some of these traits the Archaea share with the bacteria, and some of these traits the Archaea share with the eukarya. Some Archaea live in extreme environments, and they're known as extremophiles. Extreme halophiles live in highly saline environments, like Great Salt Lake in Utah. Extreme thermophiles live in very hot environments, like the Yellowstone geyser group out in Montana. Here's the heat extremophiles. It could be approaching 100 degrees in these pools here, and you can see the different colors here are different species of bacteria that are all growing in this environment, in this highly inhospitable environment. Methanogens produce methane as a waste product. Methanogens are strict anaerobes and are poisoned by oxygen. Methanogens live in swamps and marshes and guts of cattle and near deep sea and hydrothermal vents. I don't know if you guys have ever been to a real swamp, but if you get to a swamp, sometimes you'll just be sitting there on the edge of the water, and and something will bubble up just out of nowhere. That is methane that's caused by methanogens living in the swamps and marshes. Also, cows produce a lot of methane, and methane is important because it's a greenhouse gas, and the more methane that gets released into the atmosphere, the greater the exacerbation, the problem of global warming. I'm going to save some of that for the lectures at the end of the semester. Okay, here are some bacteria that grow at deep sea hydrothermal vents, and in these deep sea hydrothermal vents, these bacteria liberate sulfur, and they form the basis of the ecosystem there. The Archaean Methanopyrus candleri, which is the bacteria at the inset here, lives in the extreme heat of the black smokers, is what they call them, the hydrothermal vents on the ocean floor. And they help to liberate the sulfur from the environment there. Recent metagenomic studies have revealed new groups of Archaea. Some of these may offer clues to the early evolution of life on Earth. Prokaryotes are so important that if they were to disappear, the prospects of other life surviving would be dim. Bacteria, Archaea, they're extremely important to living systems. We need them for nitrogen fixation in plants, and they are very important to animal life as well. Prokaryotes play a major role in the recycling of chemical nutrients between the living and non-living components of ecosystems. Chemoheterotrophic prokaryotes function as decomposers, breaking down dead organisms and waste products. So without those, we just have there'd just be a buildup of useless waste. Prokaryotes can sometimes increase the availability of nitrogen, phosphorus, and potassium for plant growth. Prokaryotes also immobilize or decrease the availability of nutrients. So they can prevent when there's too much nitrogen, they can prevent an excess of nitrogen from reaching into a ecosystem as well. Okay, so here is an experiment where they grew pine seedlings, grew in sterile soils, which 3 strains of bacteria had been added to absorb more potassium than the... let me start over with that. Pine seedlings grown in sterile soils to which 1 of 3 strains of bacterium, Burkholdia glathii, had been added, absorbed more potassium than seedlings grown in soil without any bacteria. Other results not shown demonstrated that strain 3 increased the amount of potassium released from the mineral crystals in the soil. So this bacteria in the soil enabled these plants to absorb the nutrient potassium. Symbiosis is an ecological relationship in which 2 species live in close contact, a larger host and a smaller symbiont. Prokaryotes often form symbiotic relationships with larger organisms. In mutualism, both symbiotic organisms benefit. In commensalism, one organism benefits while neither harming nor helping the other in any significant way. In parasitism, an organism called a parasite harms but does not kill its host. Parasites that cause disease are called pathogens. Here is a symbiotic relationship between the flashlight fish, which is a deep undersea fish, lives in the benthic zone where there's almost no light, and a bioluminescent bacteria, which is occurring here in the patch under its eye. Right here, this is where the bioluminescent bacteria lives, and the bioluminescent bacteria gains a home from this fish, and the fish gets bioluminescent capacity. It's able to attract other fish of the same species based on the bioluminescence. Bioluminescence is the ability to produce light just based on biological factors. Okay, that's a mutualistic relationship. Both symbiotic organisms benefit. The ecological communities of hydrothermal vents depend on chemoautotrophic bacteria for energy. The best known prokaryotes are pathogens, but many others have positive interactions with humans. Human intestines are home of about 500 to 1,000 species of bacteria. Many of these mutualists break down food that is undigested by our intestines. The most prolific bacteria in our intestines is Archaea coli, or E. coli. Prokaryotes cause about half of all human diseases. For example, Lyme disease is caused by the bacterium that is carried by ticks. The Lyme disease is carried by the deer tick. Its ticks in the genus Ixodes spread the disease by transmitting a spirochete, Borrelia burgdorferi. It may cause a rash to develop at the site of the tick's bite, and the rash may be large and ring-shaped as shown, or much less distinctive. People get Lyme disease. It happens. It's not that uncommon. As a field biologist, Lyme disease was a constant concern to me. I was always worried about getting it when I was on the East Coast. It's something that's an occupational hazard. Pathogenic prokaryotes typically cause disease by releasing exotoxins or endotoxins. Exotoxins are secreted and cause disease even if the prokaryote that produced them are not present. That would be like botulism. Endotoxins are released only when the bacteria die and their cell walls break down. Horizontal gene transfer can spread these genes associated with virulence. For example, pathogenic strains of the normally harmless E. coli bacteria have emerged through horizontal gene transfer. So E. coli is a type of food poisoning, but it's also the same bacteria that's common in your gut on a daily basis. When it becomes pathogenic, that's when it has gene transfer that has enabled its virulence. Experiments with using prokaryotes have led to important advances in DNA technology. For example, E. coli is used in gene cloning. Agrobacterium tumefaciens is used in to produce transgenic plants. So there's ways that we can use prokaryotes technologically. Bacteria can now be used to make natural plastics. Prokaryotes are the principal agents in bioremediation, the use of organisms to remove pollutants from the environment. Bacteria can be engineered to produce vitamins, antibiotics, and hormones. Bacteria are also being engineered to produce ethanol from waste biomes. Okay, so here we can actually use bacteria to synthesize and store PHA, which is a compound that can be extracted and used to make biodegradable plastics. And we can also use current research that seeks to develop bacteria that produce ethanol fuel efficiently from renewable plant products. So bacteria, our understanding of bacteria can actually help us to live in the world more sustainably. Okay, here's bacteria being used for biological remediation. Spraying fertilizers on oil-soaked areas stimulates the growth of native bacteria that metabolizes the soil. This can speed up the natural breakdown of the process by a factor of 5. So this is probably Prince William Sound when they had the great Exxon Valdez disaster, and it spilled oil all over that place in Alaska, and then they sent out these groups of people that sprayed fertilizer all over the rock, and that stimulated the bacteria that then consumed the oil and helped clean up. It increased the degradation of the soil by 5-fold. Okay, here are. This is where you took bacteria and different amounts of the bacteria in different soil conditions. This is the original disease suppressive soil, which had a lot of beneficial bacteria in it. Only 3% of the seedlings of these sugar beets were stricken with fungal disease. When they killed all the bacteria by treating it with heat, then you had over 70% of those seedlings were killed by fungal disease. Okay, this is on scientific skills exercise, making a bar graph and interpreting data. This is on page 477. Okay, here is a structural diagram of a prokaryote. It shows the fimbriae, which are the little hairs-like appendages that help the cells to adhere to other cells and to a substrate. The cell wall, the circular chromosome is all bound up in there into the nucleosome nuclear region, but it's not a bound nucleus. There's the sex peleus, the flagella, internal organization, the capsule. You should review this information. It's on page 478. Okay, here is the nitrogen-fixing bacteria rhizomium. They have tested 6 different strains, and they tried to determine which strain was more effective in its symbiotic relationship with Australian Acacia. You can see that strain number 4 had the greatest plant mass yielded, and it suggests that strain number 4 is the best strain to grow that acacia. The nitrogen-fixing bacteria rhizobium infects the roots of some plants, forming a mutualism, which the bacteria provides nitrogen and the plant provides carbohydrates. Scientists measured the 12-week growth of one such plant species when infected by 6 different rhizobium strains. Okay, graph the data and interpret your graph. That's on page 480.